cardiovascular development.
We've been interested in using pluripotent stem cells, human pluripotent stem cells, as a model for cardiovascular development. And this question of heterogeneity of cardiomyocytes as they differentiate from pluripotent stem cells, this question has been the central theme of my laboratory over the past few years. The developmental hierarchy of cardiomyocytes in the context of their embryonic origin, it's not very well understood. And although we know that cardiac progenitors in vivo mature to cardiomyocytes, it's not entirely known the stepwise mechanism by which progenitors generate mature cardiomyocytes, and the commitment of the progenitors to specific subtypes of cardiomyocytes is not well known. And this is the area that we're interested. How can we use stem cells, pluripotent stem cells, to understand cardiac development in vitro? How do we, rather than just differentiating pluripotent stem cells and seeing beating cells and calling them cardiomyocytes, how can we identify the heart field specificity and also chamber specificity? And this has fundamental implication not only in regenerative medicine, but also in developmental cardiology and drug screening and also disease modeling. So in our lab, we've been interested in generating several reporter lines. These are human embryonic stem cell reporter lines that we've generated where specific transcription factors that are specific for heart fields are colored. So in our lab, we use differentiation protocols. We isolate these progenitors and we evaluate their lineage commitment and their developmental potential by performing both in vivo and in vitro analysis. We highly emphasize on understanding the structural, electrophysiological, and metabolic characterizations of these end products. So going back to developmental cardiology and understanding how the primary and the secondary heart fields develop, we hypothesized that TBX5 transcription factor is one of the markers for primary heart field. NKX2.5 is another important transcription factor in cardiac progenitors that is thought to be expressed in nearly all cardiomyocytes. There are some reports that NKX2.5 may not be expressed during development in pacemaker cells. So we generated a double reporter line with TBX5 and NKX2.5, and we hypothesized that the primary heart field progenitors can be identified by the double expression of these two markers, whereas second heart field are those that express NKX2.5 and not TBX5. So differentiating this line, we were able to isolate two populations, one that is double positive and one is single positive, and we showed that the second heart field genes are mostly enriched in the single positive population. We performed single cell RNA sequencing, and as you can see, the second heart field and the first heart field form two distinct clusters. And when we looked at differential gene expressions and doing GO-term analysis, we identified several terms that relate to structural genes, for example, filament and muscle cell development, and also oxidative phosphorylation and respiration that correspond to metabolism, and also regulation of cardiac muscle contraction or calcium handling. So going back to the isolation of these cells, we had to spend a lot of time looking at the different differentiation protocols that would yield a high population of first heart field, and we had to tweak a lot of the small molecules and the seeding density and the culture conditions. We were able to isolate a very high population of the cells that correspond to the first heart field. We looked at the gene expression and immunocytochemistry, and also we performed extensive electrophysiological studies showing that they form a homogeneous population of cells with electrical propagation, and they respond to pharmacologic therapy by increasing beating rate upon treatment with isoprotronol, and then with beta blockers we can bring it down. The same thing was done with second heart field. We were able to isolate a relatively high population of cells that only express NKX 2.5 and no TBX5 based on gene expression of some of the second heart field and immunocytochemistry, and also performing electrophysiological studies. The majority of these cells comprise of both atrial and ventricular subtypes, both in the first and the second heart field. But very interestingly, we also were able to isolate another population of cells that expressed only TBX5 and no expression of NKX 2.5. And very interestingly, we identified that these are actually nodal cells by high expression of SHOX and TBX18. And we have gone on to do some of these experiments by isolating these cells and showing that these cells are pacemaker cells and placing them on ventricular or atrial cells. You can actually pace those cells by implanting them, those in vitro. So we went ahead and looked at some of the electrophysiological and calcium handling genes, and we showed that the expression in the first heart field is significantly higher. And the comparison of the first heart field and the second heart field, we showed that it was very interestingly that as I mentioned before, they both contained both atrial and ventricular types. But recently we've been able to tweak our differentiation so we're capable of generating ventricular subtype in the first heart field and ventricular subtype in the second heart field. Therefore, we've been able to generate left ventricular cardiomyocytes from differentiating pluripotent stem cells by eliminating atrial and nodal cells. And this goes along what James was talking about previously, because we believed over the past 8 years or so that perhaps one of the reasons that we do see arrhythmias after cell transplantation is the heterogeneity of these cells with contamination with nodal cells that could serve as a nidus for arrhythmias. And as you can see here, these are first heart field ventricular myocytes, and the shape of the action potential suggests their characteristics. We also looked at the structural identity of these cells, and we showed that the first heart field cardiomyocytes, and specifically LV cardiomyocytes from ventricular cardiomyocytes from these cells, show a more mature phenotype, at least during in vitro development, to their second heart field counterparts. And this was seen by even immunocytochemistry. As you can see, even after 60 days of in vitro differentiation, the structural organization of the filaments are more organized in the first heart field compared to the second heart field. We also looked at the stiffness or the rigidity of these cells using atomic force microscopy, and looking at the Young's modulus, we showed that the first heart field and ventricular cells from the first heart field have a more rigid surface. One of the things that many people have been interested is a prospective isolation of these cells without the use of genetically modified transgenic lines. And we identified a marker, corin, that is highly expressed in first heart field cardiomyocytes based on our both bulk and single cell RNA sequencing. The first thing we did was we isolated differentiated cells based on corin, and we showed that the majority of these cells are cardiomyocytes. Then we looked at it at an organ level, just to see where it's expressed highest, and we showed that corin expression is mainly in the heart. Then we looked at the cardiomyocyte cell specificity in the heart. We performed single-cell RNA sequencing in fetal and also adult hearts, and we showed that the majority of corin expression is in cardiomyocytes. And then we looked at the specificity within cardiomyocytes in the first and the second heart field, and we showed that corin is in fact specific for first heart field. And this is based on the isolation of cardiomyocytes based on corin, showing that the majority of them express TBX5, no markers for second heart field or nodal cells. We also looked at it in left ventricular and right ventricular of both fetal and adult human hearts, and we showed that corin is highly expressed in the LV myocytes. So basically what we have done by generating a transgenic line, we've been able to isolate first heart field cardiomyocytes, second heart field cardiomyocytes. We've identified markers for their prospective isolation, and we've also been able to tweak the differentiation so that without the use of a transgenic line, we're capable of isolating ventricular versus atrial versus nodal cells from the heart field specificity. So, of course, the next thing we did was in a small animal model, we used rats, and we subjected them to ischemia reperfusion injury, and we transplanted these LV myocytes, and we performed echocardiogram after 8 weeks, and we showed that this is preliminary data, and our follow-up studies shows that there's actually a difference. What I mean by control is just differentiating cells according to the literature protocols so that there is heterogeneity of subpopulation of cardiomyocytes. Compared to LV cardiomyocytes, there is a significant difference now. I'm sorry. There is a significant difference between LV myocytes versus a heterogeneous population of cells. And when we looked at histochemical analysis, we showed that the transplantation of LV cardiomyocytes reduces the scar formation in these small animal models. We went ahead and performed large animal studies by labeling these cells with iron and transplanting them into pigs, followed up with MRI and histological analysis. As you can see, these are just some MRI images that shows that we could actually track these cells post-transplantation. And when we transplanted these cells at different stages of development, we were able to see that you can actually generate vascular cells as well, endothelial cells and smooth muscle cells. And very interestingly, the cardiomyocytes that were generated, they had very organized ultrastructure. To our disappointment, although we saw structural integration of these cells amongst themselves, we did not see any structural or histochemical evidence of any connexin 43 or integration of these cells with the host cardiomyocytes. This may be due to differences in terms of species, but again, the number of animals that we did, only 6 animals, so we're not very confident that this is universally acceptable, because I know that from other groups, when they transplanted these in pigs, they do see structural and functional integration of these cells. We don't see that. We see them integration between each other, but not with the host cardiomyocytes. So at the end, I would like to tell you that the protocol that we have generated for chamber-specific cardiomyocytes allow us not only to promote regenerative medicine by transplanting specific subtype of cardiomyocytes, but this can also be used in disease modeling and drug screening. A lot of drugs are specific to a distinct area of the heart or disease modeling when it only involves a specific chamber. Here you can see the electrical propagation was going this way. We actually put an electrode here and we overdrove it, and the electrical propagation goes there. And for those clinicians here, you can see that this is a specific presentation of fibrillation that we can actually develop with our cells so that we can actually do drug screening and disease modeling. So at the end, I would like to thank the members of my laboratory, if I can go to next, and I'll be more than happy to answer any questions. Thank you.